Thank you. All right, so my project was genetic knockout in Pseudomonas putida for increased polyhydroxyalkanoate production. So to give you a little bit of background information, right now our world is really dependent on hydrocarbons. And automatically, a lot of times when people hear hydrocarbons, they think about things like fuel for running our cars or for industrial production. But actually, one other main use of hydrocarbons is for plastic production. And plastics are very essential for numerous things. Just a few examples I have here, things like technology, cars, and being Canadians, Tim Hortons cups are obviously an essential there. So really, as our supply of hydrocarbons continues to diminish, it's really important that we come up with alternative ways to produce plastic so that we can maintain our lifestyle. And one example of a type of plastic that is thought to be a possibility of replacing hydrocarbon-produced plastic is the idea of bioplastic or biomass-based plastic. An example is polyhydroxyalkanoate, which is a type of polyester. Now, polyhydroxyalkanoates, or PHAs, are really attractive as an alternative form of plastic for numerous reasons. One of which is the fact that they do have physical properties very similar to hydrocarbon-derived plastics, and then also they are biodegradable, so that would be an environmentally sustainable option. Now, a lot of different bacteria produce PHAs, and then they store them within their cells to use as an alternative food source. So one example of this type of bacteria is Pseudomonas putida. Now, Pseudomonas, or P. putida, when it digests any kind of carbon source, for example, any kind of paper, or what I used in my experiment, octanoic acid, it produces and synthesizes PHA, and then it stores the PHA within its cell as a granule, which you can see here, which, when there are no external food sources at a later date, this PHA can then be broken down by the bacteria to be used as a backup food source, essentially. So it does this by way of two genes, the PHA polymerase and depolymerase genes. So you can see here in this diagram, the PHA-C1 and C2 are the PHA polymerase genes. So they encode for the production of enzymes that enable Pseudomonas putida to synthesize PHA. And then this red arrow here, the PHA-Z gene, encodes for the production of a depolymerase enzyme, which then enables the production of an enzyme for Pseudomonas putida to be able to break down the PHA and use it as a food source in order for it to sustain itself. And the PHA-Z gene also naturally gives Pseudomonas putida resistance to the antibiotic gentamicin, which is also important for my experiment. So the idea behind my project and the purpose of my experiment was to determine if PHA production could be increased by knocking out the PHA-Z gene. And the idea behind this is that if the bacteria is unable to break down the PHA because it doesn't have the enzyme that's necessary to do so, then this plastic will accumulate within the cell and could then hopefully be extracted for human use. So my hypothesis was that the PHA-Z knockout would increase PHA production over the period of about 100 hours. So the basic procedure I followed, first off, I had to actually do the modification of the gene. So first off, I extracted the plasmid PJQ200 from the bacteria by heat extraction. And then I used restriction enzymes to knock out the PHA-Z gene. And I have a little more detail about that later. After that, I took my modified plasmid and I used polymerase chain reaction to actually increase the quantity of the DNA that I had for the other stages of my experiment. So you see here the basic steps: denaturation, annealing, and extension. And then a little bit more detail about the gene knockout itself. So after my plasmid was extracted, I had my 3 genes on the plasmid in that sequence. I used restriction enzymes forward and reverse to knock out the PHA-Z gene. So I had 1 gene basically for each side of the PHA-Z gene to chop out that particular component. And then what I had was I just had the PHA-C1 and C2 genes remaining, but on the plasmid they were separated, so they weren't joined together. And I used ligation enzymes to join those 2 genes back together in my plasmid so you can see they're whole once again. And then in order to actually verify that the modification had been done, I used 2 particular methods. Sorry, I have a bit of a cold. The first thing I did was use gel electrophoresis to verify that a gene had actually been knocked out by my restriction enzymes. So I tested both my modified and my wild-type plasmids. I produced a gel with the ingredients you see there, and then after the cast solidified, I inserted 10 microliters of my plasmids and then also forward and reverse primers, once again to chop off on either side of the PHA-C1 and then the C2 genes. I ran my gel on 80 volts of electricity for about 30 minutes, and then you can see the result here. So the first two columns, my 1 and 2, are both wild-type bacteria. One was used as my positive control, and then the second column was the actual test for the wild-type plasmids. And then the third column you see here is my modified plasmid, hopefully without the PHA-Z gene, and then the fourth column is just the DNA ladder I compared my results to. So in the first two columns, you see that the DNA band moves about the same length within the 30 minutes. But in the third column, the DNA is able to actually move farther. And that's because without the PHA-Z gene, that there is that lighter weight, so it's able to move farther within the half hour. After that, I had to reinsert my plasmids into the bacteria. Now, any kind of reinsertion, whether it be by mini-prep or by heat shock, actually kills Pseudomonas putida, so I then would not be able to run the rest of my experiments. So what I did was I first reinserted the plasmid back into a bacteria called E. coli S17, which is compatible with Pseudomonas putida as it has similar pathways. And then to verify that the knockout had been done, to verify that it was actually the PHA-Z gene that had been knocked out and not just one of the C1 or C2 genes, I used antibiotic resistance. Now, as I mentioned in the beginning of my experiment, PHA-Z gives Pseudomonas putida resistance to gentamicin. So in my modified plasmids and my modified bacteria without the PHA-Z gene, they should not be able to survive in a gentamicin environment. So what I initially did was I separated first off individual colonies and colonies that had originated from one bacteria using a streaking method, and then I let these colonies just grow up on an agar plate. After that, I divided each of these colonies onto two separate plates. So each of these plates has the same colony, basically, that has been divided into two specific sections that originated from the same bacteria, so they do have the same properties. So if you look here in this circle, for instance, these two colonies originated from the same one. Now, if a colony is unable to survive on the plate with gentamicin, like you see here, then that means that the PHA-Z knockout has been done successfully. So I then take the sister of that colony from the plate without gentamicin and use that for the rest of my experiment, as it verifies that it did have the PHA-Z knockout. And then after that, I wanted to transfer the modified plasmids into my Pseudomonas putida. So I did that by bacterial conjugation, and you see the basic method here. So then after that, hopefully the modified plasmid would be located in the Pseudomonas putida bacteria. And then after that, I ran another antibiotic resistance test to ensure that the modified plasmid had successfully been transferred into the Pseudomonas putida bacteria. So you see, once again, if a colony was unable to survive on the gentamicin plate, that means that the modification has been done, and then the plasmid has been successfully transferred. So I'd select the matching colony from the plate without gentamicin to run the rest of my experiments. So you see, then I go to the actual PHA production assays. So I grew up my bacterial colonies in Ramsay's media with about 10% octanoic acid. Now, I selected octanoic acid because it is a refined carbon source, and Pseudomonas putida is able to readily break it down and use it as an external food source. After sterilizing my samples by autoclave, I then inoculated them with Pseudomonas putida at about a 10% concentration. I then let my bacteria samples grow up for about 12, 24, 48, or 96 hours. Okay, after that time, I precipitated out the PHAs that had been produced by the bacteria using chloroform and methanol, and then dissolved them in sulfuric acid. And then I could actually run the gas chromatograph you see here in order to measure how much of the PHAs had actually been produced. So the basic way that the gas chromatograph runs, after inserting the samples into the injector port, they'll travel through that column that you see there, and then based on the molecular mass of the specific compound, they'll travel through the column at different time periods. So based on the time at which they do arrive at the detector, it can be determined what that specific substance was. And then based on the quantity of material that arrived at that specific time, you can determine how much of a specific substance had been produced. So the basic results that I found. This first graph shows the difference in percent cell dry weight PHA between my wild type and my modified plasmids over a period of 96 hours. So you see that the amount of PHA that's produced in the percent PHA cell dry weight is actually decreasing over time, but it is decreasing more so in my blue dots here, which is the wild type bacteria. Now the reason for that is because I had a limited amount of octanoic acid food source that was externally provided to the bacteria, over the course of this time period, it is starting to break down the PHA that it's been internally producing. So the amount of PHA that's located in the bacterial cells is really going to be decreasing. But because my modified bacteria do not have the necessary genes and therefore the necessary enzymes to break down the PHA, more of the PHA is being maintained over the 96 hours. Now, I did notice, however, that the amount of PHA that was located in the cells did still decrease over the time period somewhat. So this leads me to believe that it's a possibility that there is another enzymatic pathway or another gene that's encoding for the production of an enzyme that can actually break down PHA. Now, my second graph here is just showing the difference in actual cell dry weight between my modified and my wild-type bacteria. So you see that the higher cell dry weight over the period is located in the wild-type bacteria, which again does show a positive result for what I was looking for in my experiment. Now, because the wild-type bacteria, when the octanoic acid is running out, because they still have the PHA-Z gene, they are able to enable themselves to survive off of the internal PHA source. But because the modified bacteria don't have the necessary gene to break down that PHA, the colonies do start to decline and they aren't able to survive because they aren't able to survive off the internal food source. So when the octanoic acid starts to run out, then the bacteria are going to die. And then the next graph here just shows the difference in PHA production between the wild type and the modified plasmids over a period of 96 hours. So you see, after 24 hours, there really is only about a 3% difference in PHA production between the modified and the wild-type bacteria, which is what I expected to see, because the wild-type bacteria wouldn't naturally have started to break down the PHA anyways, seeing as it has that external food source. But as the time period goes on and the octanoic acid store starts to decline, that wild-type bacteria is consistently breaking down the PHA. But the modified bacteria, because it isn't able to do so, more of the PHA is being maintained. So after 96 hours, you do see that 36% difference in PHA production, which is quite significant over that time period. So the overall results that I found in my experiment, first off, you see that the deletion of the PHA-Z gene really did increase PHA production in Pseudomonas putida over this period of 96 hours. I found that the difference between the PHA production between the modified and the wild-type bacteria really did increase and become more significant with time, to a maximum of about a 36% difference. I also noted that because of the fact that the PHA production was still decreasing over the time period, it looks like there's a possibility for other pathways that are leading to the breakdown of PHA. So some applications of my research. Now, one of the main things I considered in my experiment was the time period over which to run my tests. Now, you'll notice in my graphs, you see that the highest PHA production is after about that 24-hour time period. So you'd think that it would make sense to just stop the PHA production after that time period. But if you're looking to use this on an industrial level, it really would not be sustainable to use such a refined food source as octanoic acid. And if you are using something that's less refined, even if it be something like starch, then it's going to take that bacteria longer than 24 hours to actually break down the initial food source. So it really wouldn't be sustainable, particularly economically, to stop the experiment after 24 hours. So what you'd want to do is carry the experiment over for a longer time period and then maintain the PHA that's being produced during that particular time period. Now, another thing I'd really like to continue and extend my research with is the idea of using waste carbon sources for a food source for this bacteria. Now, Pseudomonas putida is very good at breaking down any kind of external carbon source. So things that I'd like to consider are the idea of using something like waste canola or waste starch, and then also waste starchy water from potato production, because that's a major source of waste that we don't really use for anything else. Another idea, like I showed you the picture of the Tim Hortons cup at the beginning of my presentation, Tim Hortons cups are something that could be produced from this kind of plastic, the internal lining. But without that internal lining, the paper really actually could be a good food source for this bacteria. So the idea of waste paper and waste type of plastic production that could be used to make this more environmentally friendly and sustainable. Now, obviously, this is just a preliminary stage experiment, and more research would have to be done. But right now, it does look like the idea of using genetic modification to increase the productivity of these types of bacteria could lead us to a more sustainable and environmentally and economically sustainable method of biodegradable plastic production on an industrial level. Thank you.
Thank you very much, Yama. Questions, Othello?
Okay. A couple of things. One, a general thing, and then I'd like to pursue a couple of technical things. So generally, just to give us a feel of where the ideas for this came from, on your mentor sheet that was sent around, it suggests that this is sort of ongoing research in Dr. Levin's lab.
Yeah.
Did you come in looking for a mentor, or did you have an idea that you wanted to do something along this line and sought out a mentor where you could pursue your own ideas?
Yeah, I have been working with Dr. Levin for about 4 years now, actually. So the first 3 years up till this year, what I was really working on was the idea of biofuel production. So I was kind of looking, I did do a genetic modification project in regards to that. I did go in with the idea of biofuel production, and then I was set up with a mentor through the Sanofi BioChallenge. What I was really doing was kind of studying and researching some ideas of what fuels are used for and what biofuels could replace in society. And one of the things that I did come up in my research in previous years was the idea that fuels are used for plastic production. So I kind of had this idea that if you were to delete that intermediate stage, delete the idea of fuels and delete the idea of even biofuels, and then move directly to the plastic production, that would be even more sustainable, both environmentally and economically. So I kind of presented the idea to Dr. Levin, and in his lab there are researchers that are working on things like biofuels and bioplastics. So I was working with a PhD student there, but the research that you see here is like my project that I was working on.
And technically you were able to carry out all of the steps yourself?
All the steps that you see here, I completed myself.
Okay.
In my lab book, there's all the dates that I did
So
those.
can we go back and look at one of your earlier slides, probably about 4 or 5, your gel electrophoresis? And I just wonder if you could walk us through a little bit.
Yeah.
So presumably, the DNA that was added to your gel, that was what we call loaded there, was restriction enzyme digested
Yes.
DNA.
Mm-hmm.
Okay. So could you just very briefly, in lane 1 and lane 2, you've got the plasmid with all 3 genes.
Yeah.
Yet you've got a smaller band there, which without the restriction map, you could predict that, and I'm sure that's correct. But why is the smaller band different in size in lanes 1 and 2? Can you give us a sense of that?
Yeah. Well, really what the idea of that was, with the restriction enzymes is, again, to chop the genes off on either side. And when there are almost slight errors, I guess, with the restriction enzymes, that's when you get the kind of like runoff of DNA that you see on the bottom there. So I'm thinking one possibility is because of the modification that there was, it's not entirely sure almost exactly like what base pair ends on the DNA, because when you use the restriction enzymes, there's almost a single strand that it rebonds to. So it's possibility that it wasn't like exactly specific where the restriction enzyme chops off on this gel. So it would leave that runoff there that wasn't the same as the others because it's on a different base pair.
The objective was to look at the concentration of depolymerase enzyme, right? Essentially.
Yes. Mm-hmm.
So did you actually measure depolymerase enzyme in the project?
No, I was just measuring like the function of whether it was like the end function almost, because the depolymerase enzyme, that's its purpose, to depolymerize PHA.
Right.
So
the reason for my question is if there's more than one route to get to depolymerase, wouldn't it be logical to measure the depolymerase itself as well as do the genetic knockouts?
Well, what I was kind of suggesting with the other enzyme pathway, like it's not known if there is another enzyme that's leading to that production. Like I guess what I was proposing there, not so much that there's another gene that produces the same enzyme, but it's possible that there is another enzyme that's being produced there. So that could lead to a different like pathway, basically. One thing that's really unique about PHA, it does have a lot of specific enzymatic pathways and metabolic pathways it can follow through. It usually chooses a specific one where one glucose molecule ends up being converted into one ATP molecule, or a hexose. But it is possible that there are other pathways and other enzymes that I wasn't entirely sure what they are yet. So I wasn't really sure what to look for if I was going to be doing that again. But I'd like to investigate and continue with that.
So Have you looked at longer time frames to determine whether how the survival of the bacteria were actually affected by this accumulation of the PHA? Even if you provide them with the proper nutrients, presumably they can't accumulate this forever, and it will certainly affect their survival rate. So have you looked at that at all?
Yeah. Well, the only thing I was really looking at in regards to survival rate was the cell dry weight in one of the graphs that you saw there. What you'd think on an industrial level, you would be looking to consistently giving it an external food source, so it really wouldn't affect the survival rates there. Bacteria do have a short lifespan, but at the 100 hours, I thought that was, for my experiment and for a preliminary experiment, a suitable time frame over which to run the experiment. You would think it is possible that the PHA could accumulate to a longer time period than that, which of course I would definitely be interested in researching. But yeah, there would be definitely a max at which the PHA would stop being produced, because there's limited area obviously as well.
Used gas chromatography to measure the amount of PHA that was produced, and part of your assay was to use sulfuric acid. Can you tell us what the product was after treating PHA with sulfuric acid and what you were measuring?
Yeah. Well, like what was happening initially was I was first off just extracting kind of the PHA from the plasmid, and then I was using sulfuric acid. I believe what it was an acid. I can't remember the name off the top of my head right now, but it was just like it makes it, because the gas chromatograph can't actually just read the PHA itself, so it needs to measure other products.
Has there been a lot of work done on the PHA that's derived from bacteria? It's internally located, so in this case, the gas chromatograph, obviously you use the sulfuric acid. But in terms of industrial process, extracting PHA from bacteria,
Yeah.
how much, you've increased it by 36%, is that still a commercially viable situation to use? How much bacteria do you need? What kind of processing facility would you need? Those kinds of things.
Yeah. Well, right now, there really isn't industrial production of bacterial-driven PHA. Companies have tried, I believe, to synthesize it chemically almost, because it does really have good, strong physical properties. But in regards to the bacterial production, if this were to be used industrially, I think what you need to do is continue with the increase. I think what I've done is really substantial. Rather than just trying to almost increase it immediately at like one given time point after, say, 24 hours, I think it would be, like what I've done would be really sustainable industrially, because it's over a longer extended time period, which is, I think, what really companies would be looking for, trying to maintain that so that it would be more environmentally and economically viable. I think that if I were to continue with it and to find other enzymes and genes that would lead to PHA depolymerase, or if I were to try to increase the presence of PHA polymerase, that would also help to increase the industrial viability of it.
Ella, you said you have been at this for 4 years in Dr. Levin's lab always. Is that the case? So which grade are you in now?
I'm in grade 11. Yeah, I started in grade 8 working in the lab.
Okay. And have you competed in this competition before?
Yeah, I did that. I've done the Sanofi BioGENEius challenge for the 4 years. I've actually won first place every year in my age category, but this is my first year in senior, and seniors, the nationals just for seniors. So.
Excellent.
Yeah.
Thank you very much.
Thank you.
Thanks for your time,
Thanks.
and I hope you feel better.
Thanks.